screen. Just going to add a bit one more person there that just joined us. Okay, so I'm going to now share my screen and we will get going. Before we start, and while I've got that many here, so, oh, this is not responding well. Just going to open my PowerPoint again. So while I've got you all, so many on the line at the moment, so we have 3 more lectures to do, okay? We'll do one today, then we have 2 left. The last 2 are both on the Human Genome Project. However, there is a natural break in it. And we've got a workshop to do as well. So what I thought we would do is that we will, unfortunately, I've got some marking coming in. So if it suits you guys, I would like to do Wednesday and Thursday next week. Okay, so if we could start at 8:30 on Wednesday and Thursday next week. We will go through the last 2 lectures and we will go through the workshop. So we'll maybe do a lecture on a Wednesday and on the one on the Thursday, and then we can choose to do the workshop either Wednesday or Thursday. I'm just trying to split up the lectures because I know that it can be very difficult for you to sit and concentrate for that long, really, having to listen to somebody or looking at slide and listen to somebody that is just talking without any breaks or anything, really. So I do know that it requires quite a lot of, obviously, concentration to do that. So that's why I think that we are better splitting it into one each day. We have to do the workshop anyway, so we can't finish the last in a day. We need really 2 days to do it. So from that point of view, it really doesn't make any difference. Okay, I'm now actually going to find the lecture for today because my PowerPoint have just crashed. Workshop users. Right, this one here, I think. Okay, so just give me a minute. Okay, so I have now managed to open it again. So I'll share my screen with you now. There we go. So I'll go through this lecture, and that's really all that we've got to do today. Okay, so let's go on slideshow and let's get going. So you'll recall that last week we spoke about genetic variations in humans, and we spoke about different types of genetic variation. So we spoke about mini-satellite, macrosatellite, and how these mini and microsatellite really varies between individuals in terms of how many times these sequences are repeated. So many of it is repetitive sequences. There's also single nucleotide polymorphisms. Single nucleotide polymorphisms you might very well have heard about, because that's probably the one which is most commonly used today, rather than maybe in the 80s and the 90s, people primarily used mini and macrosatellites and looked at the number of repeats. And really the number of repeats that you saw, you can use in DNA fingerprinting. So actually, this DNA fingerprint and this number of repeats varies between individuals. So although most, over 99% of our DNA is really identical between individuals. There is this a small percentage of DNA that varies and that is unique to each individual. So what we're actually going to do today is we're going to talk about how we can use that, how that genetic variation is used in mapping the human genome. And also we're going to talk a little bit about how you can locate disease genes using the genetic variation. So using these, today you would use primarily SNPs or single nucleotide polymorphisms, but at that time it was primarily mini and macrosatellites that you would be looking at. Okay, so here's the learning outcomes for the lecture. So keep thinking about these different genetic terms that we spoke about the last time to make sure that you know what they mean. Also the main topic really of this lecture today is for you to understand how polymorphisms can be used as genetic markers. And then you're going to really appreciate the difference between genetic and physical mapping strategies. So this is quite important that you're able to distinguish between what is a genetic map and what is a physical map. Okay, so we're really going back to the 80s and the 90s here, where scientists wanted to build up a map of how genes were arranged on our chromosomes, not only on humans. They actually started out with looking at other species, so mouse, Drosophila 5, worms, and those kind of things. Their genomes were actually sequenced before the human genome. However, before we move in, so this is a kind of pre-sequel to sequencing really, because before we move into sequencing the human genome, in one of the projects, what they did was really to build up a map of how the genes were arranged on our chromosomes. Okay, so what genes are on chromosome 1, which are on chromosome 4, etc., etc. And that really enabled the mapping and identification of disease genes, which you'll hear more about next year. So much more about disease genes next year. But this also really helped very much in tackling the human genome sequencing project. And this is really what we are building up to, okay? So this is the kind of pre-sequel to the next 2 lectures where we will speak in more detail about the Human Genome Project. Okay, so we're going back to your high school days now, and I'm sure you've had some genetics in high school, and you learned that some mutant traits, so if you look at the Drosophila fly, or the fruit fly as it's also called, there are some mutant traits which are linked. So for example, white-eyed mutant is linked to the sex of the fly, so male, female sex. And that means that those 2 traits do not independently segregate. So if one of them is present, then the other one is also present. You should have heard about this before. But that means that those 2 traits are linked. Okay, so they're linked together. So the likelihood of you getting one trait, if you've got the other, is really, really high. And what that really indicates is that these genes, so the genes which are coding for these traits, are in very close proximity, so close physical proximity on the chromosome, okay? So when that, when they separate during meiosis, so you should know all about meiosis, I'm not going to talk about that, but when they separate during meiosis, you do not get, or the chance of getting recombination between those two genes is very small, okay? So the closer they are to each other on the chromosome, the less likely, or the likelihood of separating through recombination is a lot less compared to if the chromosomes are, if the genes are further apart on the chromosome. And because of that, it became possible to make these very rough genetic maps of chromosomes. Okay, so this is really what is explained a little bit further here. So first of all, as I mentioned in the previous slides, it's called linkage and linkage analysis. So with linkage, you really look at whether one locus allele influences the other locus allele, okay? So does one trait influences another trait? If you have got that trait, are you more likely to also have this other trait? And linkage really is a measure of how far apart genes are from each other. Because as I said, closer to each other, close physical proximity, less likely for mitotic recombination events to occur between these two loci on the chromosome. So if you look at the illustrations down here, so that's the Drosophila fly, you can see we've got the wild type and we've got the mutant fly down here. And you can see that they've got different characteristics. So for example, the first trait here is the antennas. You can see the mutant has got these small antennas. The wild type have got these large antennas. If we look at the wings, you can see the wild type have got long wings, and the mutant has got these kind of small, quite abnormal looking wings, as you can see down here. So you can see that there's lots of traits in the Drosophila fly, and you can see that the genes coding for these are depicted along the chromosome, as you can see here. Now, for example, if we look at the antenna and the wing type of wings of the fly, you can see that those two are closely linked to each other. So those two genes are lying in close proximity on the chromosome, and recombination is less likely. Now, if we go further apart, so you can see, for example, if you look at the gene for antenna and you look at the gene for, oh, can I see it because of this one here? And you look at the gene for eye color, for example, so here's an eye color one, you can see that these are much further apart, and there is a much weaker linkage between those two phenotypes because the genes are further apart on the chromosome. So really it has to do with that when you breed, traits such as antenna and wings are closely together on the chromosome, and therefore you're more likely to see those two traits inherited in the offspring. We'll look more at that here. Now, so this is quite easy to do in the Drosophila fly. So you can do all these cross breedings between different wild type and mutant flies, and you can then look at the offspring, right? So you could breed the different combinations of wild type and mutant types and look at what traits or what phenotypes does the offspring have, and based on the phenotype of the offspring, you can then start to look at, what is the likelihood of the offspring looking like mum or looking like dad, etc., etc. And on that basis, you could start to put together a map of how close these genes that leads to this phenotype are together. But obviously you can't really do that in human, because we can't set up breeding experiments between humans with different phenotypes. And also, first of all, it would take a very long time, because obviously the gestation period is a lot longer, and ethically it just wouldn't be correct, of course, because we obviously choose who we want to marry, etc. But also, actually, in human, we don't really have enough visible traits like antennas and wings and those kind of things that makes it worthwhile to do these kind of crossbreeding experiments, really, to look at the phenotype of the offspring, because there just isn't phenotypes enough really to look at. So the question really becomes, so it might be easy to make these genetic maps in the fruit fly, because we can do different combinations of breeding and look at, what's the phenotype of offspring. But how do we actually do that in humans when we can't do these many breeding experiments? Okay, so the big idea of this, and really how they did it, so the idea really came around very much in the late 70s, as you can see down here, a meeting in April 1978. The idea of using microsatellites. So today you will probably very much use single nucleotide polymorphisms, but today at that time it was very much microsatellites that they looked at rather than single nucleotide polymorphisms, because actually the reason for it is that back in those days they weren't really aware of that there was a single nucleotide polymorphisms or that many single nucleotide polymorphisms between individuals. It was really something that they got a much better understanding of after the human genome had been sequenced. But back in those days, they were aware of microsatellites and microsatellite polymorphic locus alleles between individuals. So also at that time, the techniques were around. So they had the restriction fragment length polymorphisms, as you will recall, we looked at, where you basically chop up the DNA with restriction enzymes, and then you run it out on gel. And obviously, depending on whether you have got a polymorphism or not, or the number of repeats that you have got of a microsatellite, that will change the length of the different bands. So you will get different length of bands running out on your gel. DNA sequences, electrophoresis, PCR. So these are all techniques which were developed or were being developed at the time. And that really allowed scientists to look at polymorphisms without having to look at traits and phenotypes, but they could use these microsatellites as genetic markers. So we'll look a little bit more on that idea here, really. So this really kind of is a visual illustration of how you can use these microsatellites as genetic markers to make maps, but also really to look for disease genes. So this map here is showing you a family tree. Okay, so you can see that we've got great grandparents up here, and then siblings all the way down here. So we have got a normal male. Males, first of all, is the square, and in circles we've got females. And you can see that when we've got a white square or circle, it's a normal female or male, so non-diseased. But where it is a black circle or square, it is a diseased male or female. So it's a male or female with some kind of genetic disease. That's kind of actually irrelevant what that genetic disease is right now, okay? Okay, so in this family tree here, what you can also see is some numbers. So these numbers here, you can see there's 12 numbers, and these numbers indicate where we have taken a DNA sample. Okay, so we've taken a sample and extracted the DNA from these individuals, which are numbered in this tree here. So you can see there's a combination of male and female, and a combination of normal and deceased individuals that we have collected DNA samples from. Okay, so if you now look at the jigsaw here, so this is the DNA, and what we're now doing is that we are looking for a microsatellite or for microsatellite markers in these individuals. So we have a whole range. In fact, every single one of these jigsaws, so every jigsaw here is a genetic marker, so it's a microsatellite marker. Every single one of these jigsaws is a microsatellite marker. And every color, as you can see here, so you can see there's a color as well. So each jigsaw is a genetic marker, which is typed in each of these individuals that we've taken a sample from. So these 12 individuals that we had on the previous slide. And the color equates to the allele. And what you can also see here is in the red circle, we have got all the individuals which have got the disease, okay? That's so you don't need to go back and forward. But if you look here, so you could see, for example, number 1 and number 3 is deceased and number 2 is not. So 1 and 3 has got the circle and 2 has not, okay? So the red circle indicates a deceased member, and where there's no red circle, we have no disease. So what you really need to do now is you need to find which one of all these markers, so which of these jigsaw pieces is the piece which most accurately reflects the presence of disease. So what jigsaw marker is it that you find in every individual that has got disease, basically? Because it is that a microsatellite marker which co-segregates with disease. Somehow that marker is linked to the disease gene. So every time you inherit disease, your likelihood of inheriting or your likelihood of having that microsatellite marker is very, very high. It's actually 100% linked here. So every time you've got the marker, you've got the gene. So anybody can see which piece we're talking about here? Which one of these microsatellite markers, so that is each jigsaw piece associated with the disease? I'll just give you a wee second. I think you're all sleeping. You're not chatting either. Anybody find it? Can you see which one it is? No? So actually, you can do this in your own time, but if you look very carefully, what you can see here is that the marker, which is found in all of the individuals that has got disease, is this one down here. Okay, so if you look at that piece there, you will find this piece in every single individual that has got the disease. Okay, so I've just highlighted them there. So every individual that has got disease have got this marker here, okay, at that allele. So you can see that that particular jigsaw there is present in every deceased individual. So that really means that that microsatellite marker co-segregates with the disease gene. Okay, so they're closely linked together. Right, no, it's not moving, there we go. Okay, so in that particular example there, the marker, so the jigsaw piece, which co-segregates with the disease, so you have that marker every time you have the disease, was obviously identified. And that is really very an analog to a genetic map study. So in a genetic mapping study, what you do is you look for a marker that co-segregates with the disease gene. So you look at all these markers, you will have many, many different microsatellite markers. And what you then look at is when do these two co-segregate? So where are they linked? Which microsatellite marker is linked to the disease gene? But to build up a map, really, to build up a chromosomal location, what we really need to do first is we need to create a map of the chromosomal location. So in order to see where the gene actually is, we need a map of the chromosome, right? So that we can locate approximately where that gene is within the chromosome. So what you really need to do first, before you start looking at disease gene, is that you need to create a map of the chromosomal location of polymorphic markers. And how are these polymorphic markers positioned with respect to each other? Okay, so you can see it's quite complicated. So you have a lot of polymorphic markers. And what you really need to do is you need to build up a map of these markers to figure out, where on a chromosome is these markers positioned according to each other. Okay, so what you need to imagine, so what we're obviously going to talk about now is how you do that. So what you need to imagine is that you have identified, for example, 1,000 different microsatellite loci, and you then have the opportunity to genotype DNA for individuals to see which alleles are possessing these microsatellites. So where are these microsatellites in these individuals? Now, what you really need for that is that you need to genotype families. And at that time, it was quite common to use trios of families or extended families, so mother, father, and child. So you need to take all these microsatellite loci, and then you need to genotype all these many families. And on the basis of that, you can then start to figure out whether these alleles, okay, of two of these microsatellite loci are always inherited together, okay? You've got 1,000 microsatellites that you're looking at, and what you're looking at within these families is what is the child inheriting? So is the child, or what is the likelihood of the child inheriting two microsatellite loci? So are they always the same? Are they different? Are they mostly together, or is it a 50-50 chance? Okay, so they will inherit one from one, one from dad. And that will really allow you to see how close these two microsatellite loci are on the chromosome. So it's also called complete heritage or partial or no linkage. Okay, so complete linkage is obviously if the child always have got these two microsatellites, or it's always inheriting these two microsatellites. If there is partly inheritance, then there might have been some recombination going on, and then you can have, if these two microsatellites, for example, are found on two different chromosomes, then you would have no linkage at all, really. So they would never be found together, these two microsatellites. So let's look a little bit closer at that here, and it might make you understand it a little better. So here we've got a mom and a dad, and we're looking at two microsatellites. So you can see microsatellite 1 and 2. And this number here in the bracket shows you the number of repeat which are found of that microsatellite marker. So you can see that there's 9 repeats here, and there's 4 repeats here. So this is in mum, and of the other allele, as you can see here, has got 6 repeats, and there's 2 repeats on that one. And over here you have got dad. Okay, and here we've got the offspring. So you can see here with complete linkage. So obviously we inherited one from dad, we inherited one from mum, and you can see here that we have got complete linkage of these two microsatellite markers here, because you can see this allele here, these two are inherited from mum. You see both the microsatellite markers are inherited, from dad. Yes, from dad. And then both mum and dad has got this 9-4 here, but this one here must obviously be coming from mum, because the 3-1, as you can see here, came from dad. So the 9-4 here must be coming from mum. So you can see that here we have got a complete linkage. Okay, so there's no recombination going on between these chromosomes, these alleles, as you can see up here. Now, partial linkage, which is what you see here. So again, we've got a mum and a dad up here, and then we've got an offspring down here. Now, what is happening here is that we have got so what is happening here is that in one, so here is one offspring, and you can see that in this offspring here, we have had no recombination, so no mitotic recombination crossover has occurred, because you can obviously see here, we have got 1 here, 3-1, which is inherited from dad, and we have got a 9-4 here, which is inherited from mum. Okay, so we've had no crossover here, but you can see in the other offspring that we're looking at here, we have had some recombination going on. So again, we have inherited one from dad here, but here in mums we have had recombination going on between these two alleles, as you can see up here. Okay, so we've ended up with a 9-2 because we've had recombination up here. So that means that they are partially linked. Okay, it means they're slightly further apart on the chromosome, these two markers, than in the previous example, where the offspring, where there was no recombination at all, so there was a complete linkage between those two microsatellite markers. That is not the case here. We have got mitotic recombination or crossover really between these two microsatellite markers, so the M1 and the M2 microsatellite marker in mum up here have had some recombination going on. So really, the frequency of recombination is proportional to the physical distance that these two markers are apart. Further apart, more recombination. Closer together, less recombination. So this example here is an example which is showing you where there's no linkage, okay? And you can see in this case, showing bits of 2 different chromosomes, and it's really just to illustrate that these markers are far apart, okay? So that even on different chromosomes in this particular case here. And then you can really get any combination of these microsatellite markers in the offspring, which is what you can see down here. So independently segregation is what this is called. So these two markers are independently segregated. The inheritance of one allele at one locus really has no influences on the allele at the other locus, okay? Okay, so moving on to talk a little bit about haplotypes. So haplotype, so obviously, first of all, when you start off with doing these kind of work and maps, you obviously don't have a nice picture of mom and dad's chromosome and what is being passed on. So you're trying to build up these maps. What I'm showing you here is us already having these maps of mom and dad's. But obviously when you're starting off with this kind of thing, you don't have these maps built up yet. And what you really know is just the allele size, okay? So for microsatellite 1, we know in this case here that mom has got 6 and 9 repeats, okay? And dad has got 3 and 9, for example, here at this microsatellite that you can see there. So what you really need to construct is what we call the haplotype. So haplotype is really defined as a set of alleles of a group of closely linked genes which are usually inherited as a unit. So really, what we have looked at and what you've seen is that an individual inherits a complete haplotype from each parent, okay? So basically, these kind of maps and the hereditary pattern of these microsatellites are need to build up a picture. So you don't really know anything about this. You take a DNA sample and you obviously need to build up the picture of these microsatellites in mom and dad, and then look how these are passed on to the child. And then you need to determine, what you then determine is the haplotype of the child. And that is what you can see here. So the haplotype, what the child is inheriting of these microsatellites, really will then allow you to start building up these maps. Okay, so that's really how you do it. So you look at the microsatellites in mom and dad, and you look at how these are hereditary within the child. And based on how these are hereditary, how the microsatellites in the child, you can then determine the linkage between these markers. And what haplotypes means is it basically just it's basically just a set of them which are inherited. And if they're closely linked, they're then a haplotype. So in this particular example here, you can see that so the child has got 3 and 1 from dad. Yeah, so we've got 3 and 1 up here. So child has got 3 and 1 from dad, and it has got 9 and 4 from mum. Okay, so 9 and 4 from mum over there. So one haplotype here would be M13 and M21. Okay, So that is one haplotype which is inherited by the child. And these haplotypes are passed on tightly, so they're tightly linked. Okay, so genetics maps mythology. So how do you do that? Well, I've actually already looked at that. So you need these referent families, your pedigree families is what we use. So we used all these families, which we need to collect DNA samples from, with all these microsatellite markers, and then we look at how these microsatellite markers in mom and dad is inherited by or within the family, okay? And based on that hereditary pattern, you can then start to look at where these markers are positioned amongst each others, okay? So how are they positioned to each others? And that is just based on looking at the hereditary pattern of these microsatellites. But it's important to remember, so that is what we call a genetic map, okay? So it's a genetic map that we are building up of these microsatellites here. So basically, based on determining the relative position of these genetic markers, by looking at the frequency of marker separation during meiosis, you can make these genetic linkage maps through the generations. So really, mitotic recombination is the engine of the study. So I think that should make sense to everybody now, because I've now actually said it in kind of many different ways, but it's important to remember that this is what we call a genetic map, okay? So a genetic map basically just looks at the relative position of these genetic markers. So genetic markers in this case is what we call microsatellite markers, so these repetitive sequences. And these repetitive sequences, as we know, varies in length between individuals. So some have more repeats than others. And that's really what we're looking at, okay? Based on that, we can look at the hereditary pattern in the child through the generations. We can look at and determine what we also call the haplotype. Okay, so the first comprehensive genetic linkage map of the human genome, as you can see here, was completed in October 1992. And this is the paper, you can find that paper on MyPlace. So in this map here, it was constructed basically on the basis of 1,416 loci, as you can see. It says that at the top of the article there. You can go and read that article on MyPlace if you want. And what you see here is an example of the chromosomes from the paper. So here we've got chromosome 1, which is what you can see up here. But actually, what all these Ds are indicating down here, so all these here, these are microsatellite markers. So you can see this whole range of these microsatellite markers that has now been positioned on chromosome 1, as you can see here. And that has been done, so this is a genetic map, that has basically been done on the basis of studying these many loci and these many families, and then putting these puzzles together of how these are inherited, okay, in the offspring. So through the generation, how are these microsatellites inherited? Are they segregated? Are they co-segregating? What are they? And on that basis, as you can see here, here is a map of the whole range of microsatellites along chromosome 1. Now also what you can see here, for example,
is that you were to start looking at diseases, okay? If you were to start looking at diseases and where a disease gene, for example, is located, you can use these markers because, we'll actually see an example in a minute, but if you have built up these genetic maps, as you see here, and you can start looking at diseased families, so in families where you have had the genetic disease, can have different hereditary patterns, doesn't really matter. You could do the same thing. You would still, collect DNA from individuals within the family, and then you can start looking at all of these microsatellite markers, because now you've got some knowledge of where these microsatellite markers are, on what chromosome, in what positions, or at what loci, etc., etc. And on that basis, you can start to build up the proximity of where a disease gene might be located. So let's, for example, if you start looking at diseased family trees, and every time you have a deceased individual, this particular loci here might be coming up. Now this marker here might be coming up, suggesting that your disease gene obviously is somewhere within that proximity, so at the top of chromosome 1 here, for example. Okay, so this is really just a revision of the units of linkage measurement. So the human genome is measured in centimorgans, and what we now know, we didn't know at the time, but what we do know now is that 1 centimorgan corresponds to the recombination fraction of 1 in 100 meioses. So every 100, so 1 in every 100 meioses that occur, if you have a recombination, it is 1 centimorgan, okay? And the physical equivalent to 1 centimorgan, we also now know, is between 0.7 and 1 megabase pair of DNA, okay, depending on the recombination frequency at that part of the chromosome. So basically, we can now start, and this is important because what we're actually going to move on to talk about now is how do we go from one of these genetic maps to an actual physical map. So a physical map is a map where you are actually working with the DNA. Okay, so here's an example of how it was used in practice. So I've actually already mentioned that in the previous example, but let's have a look at it again. So how can you use the knowledge of these genetic marks or markers to locate a disease gene? Well, so as I already said, so we looked at how all these markers have been organized on chromosome 1, for example, in the previous example. So what you need to do is so first of all, you have to imagine that you now have these maps built up of all the chromosomes, okay? So chromosome 1, 2, etc., we now have these maps of microsatellite markers and where these are positioned on the chromosome. So what you can then do, as I said, is you can take samples. So in this particular case here, we have a mom and a dad. So mother is disease 3, and you can see here, mother has got the diseased gene and dad has not got the diseased gene. We're taking DNA from mom and dad, and then we're taking DNA from all of these offsprings. And you can see there's a combination of offsprings. So some offsprings have got disease, other offsprings do not have the disease, so you can clearly see that highlighted down here. Now in this particular example here, is an AP marker, so it's a single nucleotide polymorphic marker that they're using in this case. But back in the day, so that would be today, but back in the day, that would have been a microsatellite or mini satellite marker. Sorry, I just need to put on my charger on my computer. Oh, switch off in a minute, and there we go, that's it on. So what you now have to imagine is that we've got a knowledge, we've got this map of all these chromosomes, we know where all these markers are on the different chromosomes, so we know the locations of them now. So we're taking samples from mum and dad and all these siblings. We know that mum has got the disease, okay? And we can actually see here in the offspring down here, we can see when we start to look, so we're finding the marker which is co-segregating with the disease, okay? So you can see here that in the diseased children, and so here is one with disease, there's one with disease, there is one with disease, and there is one with disease. So you can see here that in 75% of cases, so if you look at all the deceased, you can see that in 75%, the marker is present, and if you have the disease. Obviously, you can see in this individual here who is deceased, so have the disease, that child does not have the SNP marker, so the marker on that copy of the chromosome, which in most instances are co-segregating with the disease. You can also see that the marker is not present in children which do not have the disease. So really on that basis, so when you've got a picture of where that marker is, you can then start to zoom in on where or what might be the defective gene. So what is the defective gene that is causing this disease? So based on us having this genetic map of where that marker is, we can then start to zoom in and or home in on what genes are present at that proximity or location of the chromosome. So back in the day where we didn't know and had the location of all the genes, that might require obviously that you would start to sequence that particular part of the chromosome, okay? And that would then lead to perhaps a number of different genes being identified. But we will talk more about that actually next year. So next year you'll be looking much more at disease genes. And also you will actually next year be looking much more at how we used or how they used this to locate the genes which were responsible for cystic fibrosis and the BRCA gene, for example, which highly increases mutations in the BRCA gene, highly increases the chances of breast cancer, for example. This is something we'll look much more at next year in BM321. Okay, so obviously, as I've already alluded to, is that genetic maps do obviously not give you a physical map. So genetic mapping is obviously very much a mathematical exercise. So you've got all these markers and then you sit and find all the patterns, hereditary patterns, and these polymorphic markers. So really, it only provides a very rough idea of where these markers are and where the disease genes is located. So is it at the top of chromosome 1? Is it the middle? Is it the bottom? Is it, you know, it gives you a proximity of location. But eventually, of course, you will have to make the transition to working with actual physical DNA. So if you know approximately where your disease gene is, okay, you can then clone it, and then you can start identifying it. I've already said that we'll hear more about that in BM321 next year. What we want to do now is that we really want to use that information to start the human genome sequencing project. So making these genetic maps and then progressing that into physical maps was actually really, really important for the human genome sequencing project. Okay, so how do you make a physical map is what we're going to look at in the last bit of this lecture today. I'm just going to look to see if you've asked anything. No, you've not asked any questions. So what we're going to really look at now in the last bit of the lecture is how do you now convert this genetic map into a physical map? So how do you collate, organize, and distribute huge quantities of DNA to make a physical map? Well, you need to make a library. Okay, making a library involves obviously the ability to store. You need to store the DNA. You need to be able to duplicate the DNA because you're highly unlikely to have quantities enough to do everything that you might want to do. And also, it would be very nice, and you will see that certainly was done during the Human Genome Sequencing Project, that you can distribute some of the DNA. So when we come to talk about the Human Genome Project, really clones or DNA was sent out to, there was many collaborators all over the world. It was initiated in the United States, but many scientists across the world really participated in this process. So we need a library where we can store all this DNA, where we can duplicate and distribute. We also need a database. So we obviously need a database so that we can start looking at the relationship between all of these markers and all of these clones. So I've said clones quite a few times here, and you might not know what it is, but we'll get to that now. So why do we need to clone DNA into genomic DNA libraries? So that's what we need to do now. So basically, you can't work with the whole genome because it is just far too cumbersome and complex to work with. So you can't just take the whole genome for an individual and start working with it. It's just too big. So what you do is, and it's also limited in quantity. So I've already kind of mentioned that. So we need the ability to be able to duplicate it. So what you really do is that you chop up your DNA. So you take your genomic DNA and you chop it up with restriction enzymes. So you cut it into pieces. You then take those pieces and you put them into plasmids. And these plasmids will then allow, they're kind of like, as you can see here, photocopies. So they will replicate that bit of DNA. And what you can then do is that you can have many of these clones which are the same, and you can share them with your colleagues. What you also need to do, of course, it could be, particularly when we didn't know the human genome sequence, you have to think about, now that we know it, it's a different story. But you have to think about that initially we did not know the sequence of the human genome. So obviously taking some DNA and just chopping it up would not allow you to know where did that piece of DNA actually come from. And particularly as we know there's so many repeat sequences in the genome, it could make it quite difficult to determine exactly where a clone is. So what we need to do first of all is we don't need only to chop it up, we need to chop it up, we need to put it into vectors or clones, and we then need to assemble them. So we need to rearrange all these clones in to the order, the correct order, okay? So chromosome 1, 2, 3 would be a way of arranging it first, but then we need to arrange them along the chromosomes. So this is really where the genetic mapping, of course, comes along, because we now have all these markers and we know where these markers are positioned. Okay, we know where these markers are positioned in the chromosome, so we can really use them to organize these clones. Okay, so what do we clone them into? Well, you all know probably, I assume that you already know this actually. So you know that bacteria has got plasmid, so that's the genetic material that they carry. So this is circular, plasmids are just circular DNA. And what you can do is that you can take these plasmids and you can insert foreign DNA into these plasmids. So basically, you'll see in the next slide, you just use restriction enzymes to chop the plasmid, to chop the DNA, and then you can actually insert that bit of DNA into that plasmid. And that basically means that the bacteria become photocopiers, because it's bacteria. You can then transfect bacteria with these plasmids, and the bacteria will then start to copy or to produce the DNA as they proliferate. So, you know, bacteria grows really fast, and as they're growing, obviously the DNA and that plasmid becomes copied. So that means that you can get quite large quantities of DNA, and you really need large quantities to be able to do sequencing, particularly at this time. So this might make more sense to you when you're looking at this image here, because what you can now see is we've got your plasmid up here, okay? So this is a plasmid, which comes from a bacteria. And what you've got in that plasmid, you've actually got inserted an antibiotic resistance genes, okay? That becomes important for our selection later on. But for now, if you just know that in that plasmid, we have transfected the plasmid with this gene, which is an antibiotic resistant genes. What you then do next is you take a restriction enzyme. So here's EOCA1, okay? Here's our DNA, okay? So here's our genomic DNA. We caught the genomic DNA with the same restriction enzyme, which you can see here. We get those sticky ends. So remember, when you're looking at the cut, so restriction enzymes cut at a specific sequence, but not all restriction enzymes, but this restriction enzyme, ECO1, does these hangouts, so overhangs, okay? And obviously we know the sequence of that overhang, because the sequence of that overhang is obviously based on where the restriction enzymes cut. Anyway, that really means that this bit here, which has been cut with ECO1, so all the genomic DNA has now been cut up, and all the ends here will insert into the plasmid, because they will be base pairing. Oh, somebody's drawing on the PowerPoint. They will be base pairing with the plasmid here. Okay, so we now got genomic DNA inserted into our plasmid down here, basically just from cutting with the restriction enzymes and then getting base pairing. What we then do next is that we have bacteria. So we've got some bacterial cells growing in a culture down here, as you can see. We add our plasmid, we transfect, this also is really what we call it. So we transfect our bacterial cells with the plasmid. And some of those bacterial cells will take up that plasmid, others will not, okay? So this is where the antibiotic resistant genes now becomes important, okay? Because remember, our plasmid does not only exist of our inserted genomic DNA, but it also consists of this antibiotic resistant gene. So what we can now do is that we can now treat our bacteria with antibiotic, and only the ones which have got this plasmid here inserted will survive, because they're resistant to the antibiotic. All the bacterial cells which have not been transfected with this particular plasmid here will die, because they're not resistant to the antibiotic. Once we've done that, so once we've selected for our plasmid, we can obviously then purify that DNA. Okay, so we can now purify, as you can see down here, we can purify the DNA. Okay, so this is really just repeating that again. So what do we do? Well, to make a DNA clone library. Well, what we do is that we take some human genomic DNA, we cut it into smaller sizes or smaller chunks by using restriction enzymes. We then take a plasmid, we cut the plasmid with restriction enzymes, which will allow us to insert that genomic DNA into that cloning site in the plasmid. That then allows us to select and replicate for the DNA. So I'm not going to go into detail, but obviously, as I say, it's based on that antibiotic resistant genes. So once we have replicated and made lots and lots of this bits of DNA that we have inserted, then we can obviously store it as a clone library. And one single bacteria with one of these bags in it will go into a clonal population or colony. So it will grow into exactly the same as its ancestors, really. You can obviously then store it in a freezer, and you can take it out whenever you need it and to do whatever. And you can make a cDNA library, which then presents all the genes which are expressed. So remember, mRNA is really the expression of the genes. This is really just repeated again here, how you make a clone library. So you can see you've got a bit of genomic DNA. You cut it with restriction enzyme. And you get all these fragments, and you get overlapping fragments, as you can see here. And you then clone that into bacteria or yeast. I haven't really mentioned that, but into bacteria and yeast. You then propagate it, and then you can basically just store it in the freezer, as you can see here. Now, the next question is then, how do you then turn that library into a physical map? Because at the moment, all we have got is all these back clones. So now we need to turn it into a true physical map. So for that, we will use our markers from our genetic map. So we'll take markers from our genetic map, and we can start to find out which clones contains what genetic markers, and that will then allow us to order the clones, and it will also allow us to find overlapping clones. So on that basis, we can now start to build up an actual physical map of what the chromosome or what the genome is looking like. I'm not going to talk too much about chromosome walking in this particular talk, but chromosome walking is a technique that you can really use to look at overlapping clones or overlapping pieces of DNA. We'll talk more about that, I think, in the next lecture. Okay, so on the basis, now we've got all these clones, we have used markers. We've used these markers, so we've stained bits of these different pieces of DNA that we have cloned to figure out how we can organize this from a genetic map, really. So remember, our markers are very much initially based on genetics, but we're now converting it into a physical map. So we're using those markers to make this physical map that you can see here. And this is really just an example. So when you make this physical map, it's called a contig. And a contig is basically just a virtual stretch of genomic DNA, which is made up from the analysis of multiple overlapping DNA clones, okay? So we've got all these DNA clones in the freezer, we're looking at these microsatellite markers. So every single, so see these symbols here, the circle symbol. The circle symbol is a microsatellite marker, okay? So what you can see, if we take this clone here, for example, okay, B1, so you can see that that clone is found in all, or sorry, that microsatellite marker is found in all of the clones that you can see here. Oh, excuse me. And the same, you can take any other. So all of these are very microsatellite markers. So here you can see, for example, we've got two clones. So this A4 marker here is found in this clone here, and it can be found in that clone there. And you could go through the whole map, but basically you take all of your DNA clones. So you've got genomic DNA, you've used all these restriction enzymes, and you have used your markers, you've organized it, and now you're trying to make these contexts to see where you've got overlapping clones. And that's basically what you see here. Okay, so eventually the complete genome can be constructed in the clones. Okay, so you start then organizing them. So based on our genetic map, we know where these different microsatellites should be in a chromosome. Okay, we know the order in which they come based on the genetic map that we've already made. And we're now converting that into a physical map. So now we've got actual physical DNA that we're working with, and we're now looking for those markers within our DNA library. Okay, so we'll clone this into all of these bacterial clones, and we're now figuring out what markers do all of these individual clones contain, so that we can start making convert that genetic map into a physical map. And then we can obviously start studying each clone in detail. And on that basis, you also would then have access to the precise chromosomal DNA stretch that you might be interested in. So we talked about already how you might have identified a gene within a certain region of the chromosome. And obviously, based on the map that we've just built of our physical DNA, you could then go in and you could find, okay, so we have located this disease gene to chromosome 1 approximately in this location here, because it was always co-segregating with this microsatellite marker. We have organized our physical map according to that. So clone number 23, we know contains that microsatellite marker that is always co-segregating with disease. So you can go in and find clone 23, and you could start looking closer at all of the, of course, not only one clone, but you could start looking closer at all of the clones which contains that particular microsatellite marker. And you would then know that your disease gene would be somewhere close to that microsatellite region. And actually, that is a way in which we have identified a lot of disease genes. But as I say, we won't talk. We'll talk more about that next year. And it is also very convenient for the human genome to have the whole genome ordered in that sequence. And that's something we'll talk about next time as well, because that becomes particularly important actually, because there's so many repeat sequences in the human genome. So actually being able to order it, you will see, was quite an advantage. So that's just really the last slide. So what we'll talk about in the next lecture is now how we start to sequence all this DNA and how they went about it. But really, before they started the Human Genome Project, they had to build up these genetic maps and physical maps. And we'll look at the next time what they then did after that. So there's just a summary. So really what we do is we use these polymorphisms as markers for mapping. You should know the difference between a genetic map and a physical map, so that's quite important. I don't know why it says LOD scores here, but you're not to really worry about that now. But as I said, you build up these genetic maps. These genetic maps are very much based on the hereditary patterns, so build up of family trees, pedigree families, looking at the hereditary patterns of different, in this case, particular microsatellite markers. And on that basis, then starting to construct a genetic map of how these are placed in relation to each other, again, or along the different chromosomes. Going from genetic mapping to physical mapping. So physical mapping is where we've now actually working with, we've got some DNA and we need to align that. We need to make a physical map of that actual DNA. And the way we do that is really through alignment of genomic DNA library clones into a context. So you take DNA, you clone it, you make lots of copies of it, and then really based on markers from your genetic mapping, you start making your physical map. So I hope that has made perfectly sense to all of you. Have you got any questions? If you don't have any questions, I'm really happy to see how many has turned up. That's a pretty good turnout, 50 people. But nobody has said anything today, not a thing. I'm thinking that you're all sleeping because you've not even said anything in the chat. And so I am wondering whether you're actually all very sleepy or whether you're all sleeping because you've been too busy and you're all tired because you've been traveling yesterday. Anyway, I'm going to leave you, and just a reminder, so we have got next week, we have Wednesday and we have Thursday, okay? We will start 8:30 as we've done today. So 8:30, and I will be posting, probably today, on WeChat. I will post you the workshop, okay? So in the workshop, we're very much going to be looking at DNA and primers and that kind of thing. But what you need to do for the workshop, so there are some questions and you need to go through those questions for a workshop. Yeah? Everybody happy with that? Right, so 9 means the number of repeats. So it's microsatellite marker 1, okay? So sorry, somebody has asked the question here if you don't understand what I'm talking about. So M1, so it's microsatellite marker number 1, and it means that there is 9 repeats. So if you go back and you remember that each individuals have got different repeats of these markers. So it's this repetitive sequence which repeats itself over and over again. And in this particular case, there's 9 repeats of that sequence. That's what the bracket and the 9 in the bracket means. Anybody else wants to ask anything before you go? Are you all very hungry? Desperate to go for your dinner? Yeah, you're definitely all very quiet. Anyway, I'm going to let you go, okay? So please look at the workshop before next week, and I'll put it up on WeChat for you. So see you all very soon.
Bye-bye.
Bye.
Bye.